The following film is brought to you by L&J Productions. This is Gina. Gina lives on a farm in Pennsylvania. Today, we will be looking at one of Gina's diploid cells about to enter meiosis. You'll notice that meiosis 1 is labeled in the dark blue, whereas meiosis 2 is labeled in yellow.
This particular cell is about to begin prophase 1, so let's take a look. The genetic information is jumbled in the nuclear envelope of the cell. This is called chromatin. The centrosomes are also formed towards the top of the cell.
The current state of the cell, interphase, is a time for the DNA to replicate and the diploid cell to prepare for meiosis. As this cell moves from G1 into S phase, the chromatin is preparing to be copied. Interphase is not needed in between the two meiosis cycles because DNA has already been replicated, and it doesn't need to be replicated again. However, interphase at the beginning of meiosis 1 was necessary due to DNA replication.
Gina's cell now has 8 chromosomes. This genetic information is needed for the division of the cell later on. In G2, the cell is growing, developing, and preparing for meiosis. You can see that the cell is now at the start of prophase 1. The chromatin has condensed into chromosomes, and the chromosomes are visible. These chromosomes are referred to as duplicated chromosomes or homologous chromosomes.
Meiosis uses homologous pairs of chromosomes because they have different genes for the same inherited traits, whereas homozygous chromosomes contain the same genes for the same traits. This would deem crossing over useless, and genetic variation would not exist. The spindle fibers are forming in the cell, and the nuclear envelope has dissolved.
Crossing over, or the exchange of genetic information in the chromosomes, is now taking place, which concludes prophase 1. Moving into metaphase 1, the chromosomes will line up horizontally along the metaphase plate in the cell. The spindle fibers are attached to the chromosomes now, and the nuclear envelope has dissolved in order for the cell to later expand.
Transitioning into anaphase 1, the sister chromatids are now being pulled apart, and the microtubules are shortening. As the plasma membrane starts to expand, the cell enters into telophase 1. In telophase 1, the chromosomes are still duplicated and will remain that way until telophase 2. However, in telophase of mitosis, the chromosomes have separated and are ready to divide into 2 different cells. The meiosis cell has double the amount of chromosomes. The cell continues to stretch until we are in cytokinesis. 2 separate nuclear membranes form, and the cytoplasm divides, forming 2 haploid cells from the 1 diploid cell.
Chromosomes are reduced during meiosis 1, from prophase 1 to telophase 1.
In prophase 2, the 2 haploid cells that resulted from meiosis 1 are now undergoing the meiosis 2 process. In these 2 cells, the spindle fibers are beginning to form, and the chromosomes slowly begin to venture towards the metaphase 2 plate. Now that prophase has come to a close, metaphase 2 has begun, and the chromosomes are aligned vertically along the metaphase 2 plate, which is different than the metaphase 1 plate in the sense of direction.
As you can see, these chromosomes that are lined up are not genetically identical because the crossing over in prophase 1 creates genetic variation. Meiosis yields genetically different cells, whereas mitosis yields genetically identical cells. The spindle fibers are now attached to the chromosomes to ensure the separation of sister chromatids in anaphase 2.
In anaphase 2, the sister chromatids are being pulled apart in opposite east and west directions of the cell. The cell begins to stretch, and now entering telophase 2, the nuclei begins to form around the chromosomes, and the cleavage furrow begins to develop.
Now entering cytokinesis 2, the cleavage furrow develops and eventually causes the 2 plasma membranes to pinch off and divide. The chromosomes are reduced, thus resulting in 4 haploid cells that are genetically different from each other. That concludes meiosis 2 as well as the meiosis process.
Thanks for watching our meiosis production.
And special thanks to Jim Bob, Gina's owner, for letting her participate in our production.